Hello dear students, let me start the third lecture about the digestive system, which deals with glands which are associated to the digestive tube. You already know salivary glands, which belong to this group because they were part of the first lecture about the digestive system. So today I will speak only about pancreas, liver and bile ducts, which join liver and intestine. Pancreas is a large gland situated in the retroperitoneal space of the abdominal cavity, which is composed of two portions. There is exocrine and endocrine component of this organ. Exocrine component, which I'll take as a first, is, as you can read in this slide, a compound alveolar or acinar gland with purely serous secretion. This is a typical classification of this gland. You should every time know the classification of any glands which you are describing or which you are trying to recognize in the histological specimen. So in the case of the exocrine component of the pancreas, it is compound acinar gland. Compound means that it has several levels of duct system, which is very highly branching. Alveolar or acinar means that its secretory portion is composed of spherical structures, which I am now delineating. Spherical structures called acini, and no other secretory parts are here, and pure serous. It is more functional, their morphological classification. It means that the secretion of exocrine pancreas is liquid with low viscosity and with no contamination or no mucous component. Serous acini are composed of serous cells. Serous cells are the cells with very typical structure. You already know them because there was a discussion about them in epithelial tissue, or we were studied them in some previous glands. They have pyramidal shape. For example, here you can see the shape of the cell, or here the borders of this cell are well visible. Nevertheless, there isn't nucleus visible, but here is a typical pyramidal shape of a serous cell. Their nuclei are situated somewhere between the center and basal surface. Nuclei are spherical. Their content of euchromatin is quite high because the level of proteosynthesis in these cells is very, very prominent. The cytoplasm is highly basophilic in the area around the nucleus. You can see in all these cells here violet color under the nucleus or around the nucleus. So perinuclear cytoplasm is highly basophilic. It is because there is a rough endoplasmic reticulum accumulated. Rough endoplasmic reticulum is necessary for the production of proteins in these cells. These proteins are then accumulating in granules. You can see these granules in the apical part of cells. They are quite small, nevertheless visible in the light microscope. They measure about 1 micrometer, and in this case these granules are eosinophilic. You will see in one of next pictures what a huge mixture of enzymes can be found in the granules of pancreatic acinar cells. The pancreas is, or the secretion function of pancreas is regulated by many factors, many hormones, and it means that in any moment the cells don't secrete all the hormones, or sorry, all the proteins, all the enzymes together. That sometimes some enzymes are secreted more and another less, and this means that the tinction of granules can be very variable. Sometimes granules are eosinophilic, as in this case, you can see in the photograph, but there are situations that the enzymes have another composition, and the overall tinction of granules is basophilic or something between eosinophilia and basophilia. It is really very variable in the pancreatic acinar cells. Nevertheless, the perinuclear basophilia is stable feature of the cells. If you look at the same cells under the electron microscope, you will see the same things but in much higher magnification. Nucleus will be again spherical, light, with prominent nucleolus here. There will be again rough endoplasmic reticulum around the nucleus, but in this case there isn't any basophilia, but you can see parallel cisternae of this rough endoplasmic reticulum. If you see such parallel, or here it is very visible, if you see such parallel cisternae in any cell, it is a proof of a very high activity of rough endoplasmic reticulum. Golgi complex is also very prominent. It is situated between nucleus and the area where granules are accumulated. You can find components, that means small cisternae and vesicles of the Golgi complex in this area above the nucleus, and here in the apical parts, which are equipped by several small microvilli, are granules. Serous granules are electron dense, spherical, with no tendency to fuse. You can see that the granules here are called Zymogen granules. It is a very special term which is used only in the pancreas. Zymogen means inactive enzyme. Also, you can find the name proenzymes for this component. Proenzymes or zymogens are stored in the pancreatic serous granules because it would be dangerous to activate the enzymes too early. They should be activated after the secretion to the intestine. If activated too early, they can digest the cell itself, and it can be very dangerous. It can be very serious disease called acute pancreatitis, leading to very serious problems or death in very short time. So to prevent pancreatic disease or acute pancreatitis, the enzymes are inactivated and protected against preliminary activation in the granules. Higher magnification of the same granules doesn't show anything new. Rough endoplasmic reticulum in this picture is more like vesicles than cisternae, but that's only because the cisternae are here in this picture cross section. You every time can recognize rough endoplasmic reticulum according to ribosomes which are attached to its outer side of a membrane, and I think they are visible here. Pancreatic acini are not empty usually. The luminal cavity here is filled by small cells which are called centroacinar cells. It is a very special feature of the pancreatic acini, and there is possible to recognize the pancreas according to presence of these cells inside. If you compare this picture with the same area of some salivary gland, you would find here free space. Lumen of the acinus doesn't contain any cells, but in the pancreas you can find them here. Of course, not in every section. If the section isn't through the luminal or through the central area of the acinus, you don't see any pancreatic centroacinar cells. But for example, here it is here one, or I don't see any other here. But the section really must go directly through the center of the acini, because the lumen of the acinus is usually very, very small. These centroacinar cells are not secretory cells. They don't secrete any proteins. They in fact belong to the system of pancreatic ducts. They are situated as blind ends of the smallest pancreatic ducts, which are connected to the acinus, and which you can see here, or here is another section of such duct. These ducts, these smallest ducts, are called intercalated ducts, and they are really intercalated to the interior of the acinus, and their first cells represent centroacinar cells, like you can see in this schema. Intercalated ducts are present in salivary glands as well, but in salivary glands they begin at this level. They begin at this level and they don't penetrate to the lumen of acinus. Nevertheless, here in the pancreas they do it, and so the lumen of the acinus is full of small centroacinar cells, and the secretion of pancreatic acinar cell has to sometimes penetrate through the intercellular spaces between these centroacinar cells to reach the lumen and to be collected by a duct system. The same situation can be visible also in the electron micrograph. Here is the apical portion of acinar cell. You can recognize it according to serous granules and rough endoplasmic reticulum. And here are several microvilli which are situated on the apical surface, but you can see that these microvilli are deformed. They don't stand, they are not free, because there is no space. Instead of free space here, centroacinar cell is here present. Sorry, I don't cross it, I just want to underline it. Centroacinar cell is here present. You can see that it is a cell having very, very pure cytoplasm, where some mitochondria are present but no other typical organelles. Ductile system or duct system is beginning by these centroacinar cells, and then goes on as intercalated duct. As you know that the exocrine pancreas is compound gland, you probably expect some higher levels of the duct system, and really there is a level of interlobular ducts, then interlobular duct, and then main duct. Probably you know that the pancreas has one main duct and one accessory duct. There is a rule that the main duct of any big gland is lined by the same epithelium as is in the organ where does it go. The main and accessory duct of the pancreas go to the duodenum. Duodenum is a part of a small intestine which is lined with simple columnar epithelium. So the main or accessory ducts of the pancreas are lined also by a simple columnar epithelium. No higher. There isn't any stratified epithelium as you can find in the big ducts of the salivary glands. And that's why the change of epithelium in the duct system can be much slower than you know from salivary glands. Intercalated ducts are lined by simple to simple. Of course, all epithelia are simple, but in intercalated ducts, the epithelium is simple squamous to cuboidal. Then in intralobular ducts, it's cuboidal. Interlobular ducts can be lined by simple cuboidal to columnar epithelium, and the main duct or accessory duct are lined with simple columnar epithelium. Here are several pictures showing these duct levels. So we are starting once more in the lumen of the acinus, where are several centroacinar cells. By the way, in this case, I think you can see it, pancreatic serous granules are basophilic. As I said, the tinction of these granules can be very variable, sometimes basophilic, sometimes eosinophilic. It depends on the combination of enzymes which are expressed at the given moment. But let's go back to ducts. Centroacinar cells then line the intercalated ducts, or maybe better is to say that the intercalated duct is lined by the same squamous cells as are the centroacinar cells, so it is still intercalated duct. You can see blood vessel near the intercalated duct. You can see that the epithelium of intercalated duct is quite similar to the endothelium of a small blood vessel, but it isn't anything surprising. Both are simple squamous epithelia. When you go to the bigger ducts, for example, here it's possible to see as the simple squamous epithelium is gradually increasing its height. Here it's still squamous, here it's cuboidal. You can see that the cells have the same height and width, and mainly the nuclei become spherical because they correspond in shape to the shape of whole cells. So intralobular ducts are lined with squamous to cuboidal epithelium. Interlobular ducts, which are situated in the septum between lobules, and so which are surrounded by more connective tissue, they have simple cuboidal epithelium. And here is some bigger, larger interlobular ducts, where epithelium is in one side still simple cuboidal, but here in another side maybe columnar, simple columnar epithelium is developing gradually. So yes, the interlobular ducts, which are surrounded by a high amount of connective tissue, are lined with simple columnar epithelium. Here is a picture which I promised a while ago. What are the enzymes secreted by pancreatic acinar cells? You can see that there are very many. Trypsinogen or chymotrypsinogen, carboxypeptidase, all these three enzymes digest proteins and peptides. Ribonuclease and deoxyribonuclease, they digest nucleic acid. Triacylglycerolipase or phospholipase, they digest lipids. Elastase, it is special protease, so it belongs to the group, to this first group. Amylase is the enzyme digesting polysaccharides, starch or glycogen. It depends on the origin of this polysaccharide. So you can see that all nutrients can be digested by pancreatic secretion, and it's logical that no cell can produce all of them together. It depends on the composition of the eaten food, which is recognized by special chemoreceptor cells in the intestine. You probably know the tuft or brush cells in the intestine. Intestinal enteroendocrine cells secrete some hormones which are influencing acinar cells, and the resulting composition of the secretion depends on the equilibrium between several hormones secreted by enteroendocrine glands or enteroendocrine cells. Here are just examples. Secretin is one of these hormones. It is a peptide secreted by S cells. Its high level triggers the pancreas to form a high amount of secretion, which is quite poor in enzymes. On the other hand, cholecystokinin is another hormone secreted by I cells of the intestine. It stimulates pancreas to produce more enzymes, and the secretion becomes more viscous. I skipped the first row in this table. The exocrine pancreatic secretion has to contain also water and ions. Water is logical, it's fluid, so these enzymes must be soluted in something. Ions are also necessary here, and mostly bicarbonate ions are important. Bicarbonate ions are transported by cells of ducts. Also centroacinal cells can do it, and their high level is important for the neutralization of acidic secretion or acidic juice coming from stomach to the intestine, to the duodenum. So this acidic stomach juice or gastric juice has to be quickly neutralized, and this neutralization is here the cooperation of the alkaline secretion from pancreas and also from Brunner's glands in the duodenal submucosa. I think that I can finish the exocrine pancreas, and let me start the endocrine component of the pancreas. Endocrine component is a collection of many Langerhans islets. Langerhans islets are microendocrine glands. Their number is very enormous. That's possible to find about 1 million of islets in one pancreas. Their size is very variable. This one here is quite big. It can measure more than 200 micrometers in diameter and is composed of many cells, but there exist also micro islets composed of several cells only, so very variable size is here. If you compare cells of the islet with the cells of the exocrine pancreas, you can see that they are less basophilic. Their cytoplasm is only light basophilic, almost homogeneous. We don't see any granules here in the electron microscope. Nuclei are smaller and darker compared to cells of the exocrine pancreas, and also arrangement of the cells is different. These cells of the endocrine pancreas are arranged in chords, and so they form trabecular epithelium. So the Langerhans islets are composed of trabecular epithelium. Trabecular epithelium is never alone, it must be supported by some stroma. The stroma of Langerhans islet is composed of reticular connective tissue. So you can see some connective tissue components like here or here among the islets. Some collagen is here. Of course, it must be collagen type 3. The capillaries are situated in this connective tissue stroma. Capillaries of Langerhans islets are very important because the secretions, the hormones of Langerhans islets are secreted to these vessels, and so they are special. They have fenestrations in their wall, so capillaries of Langerhans islets are fenestrated. So there are fenestrated capillaries, reticular connective tissue, and the main parenchyma of the islet is trabecular epithelium. In this staining, hematoxin and eosin or another synoptic staining, there isn't possible to distinguish any types of epithelial cells here. All the same seem to be the same. All the cells here are secreting polypeptides because all hormones of pancreas are polypeptides. This picture is special histological technique showing vessels, so you can see a rich capillary network in the pancreatic stroma in this, or Langerhans islet stroma in this picture. Here is a high magnification of hematoxin and eosin staining where you can see directly capillaries. Here is a cross section, here is a longitudinal section of a capillary. You can see endothelial cell. Of course, it isn't possible to recognize whether this capillary is fenestrated. You need electron microscopy to find this. Again, as it is synoptic staining, all the cells here seem to be the same. Nevertheless, when you use some special staining, like for example aldehyde fuchsine, you will find some cells blue and other cells red. So there is a, sorry for the low quality of this picture, but I haven't got any other. Some cells are red and other cells are blue, and there is also the third population of cells which don't stain well by aldehyde fuchsine. So according to this staining, it seems that there are three populations of cells here in this islet. If you use immunohistochemistry, you can find more cell types. It was discovered that there are 4 main types in the islet of pancreas or islet of Langerhans, and several less frequent minor cell populations, but 4 main cell populations. The most frequent cells of the pancreatic islets are B cells. B cells or beta cells, they represent approximately 70% of cells in the islet parenchyma. They are situated mostly in the center of the islet and they are accumulating in clusters. We know that beta or B cells secrete the main hormone of the pancreatic islets, which is insulin. So insulin, I hope that all of you know this hormone, because it is very important. The disorder of its production is diabetes mellitus, so it is secreted by beta cells. The second frequent are A or alpha cells, which represent about 20% of cells here. They are situated mostly in periphery. They secrete glucagon. Glucagon is another well-known, another well-known another well-known hormone of the pancreas, glucagon. I must write it. Pen doesn't work. Now it will be KIO glue. I am not in the picture. Glucagon. Okay. Glucagon is a hormone with opposite function compared to insulin. Insulin is decreasing the level of plasma glucose because it stimulates cells to transport glucose through the membrane into the cytoplasm. Glucagon has the opposite function, and it increases the extracellular level of glucose. About 5% of cells in the pancreatic islets are D cells, which secrete somatostatin, a hormone which you already know from some enteroendocrine cells. These cells are scattered in the islet. They haven't got any prevailing or privileged position. Approximately the same number, that means about 5%, is represented by F cells or PP cells, which secrete pancreatic polypeptide. All these cells can be recognized by immunohistochemical proof of their products. So everything what you need is a good laboratory and antibody against any hormone. So for example, if you use an antibody against insulin, which you can buy this antibody, you drop it to specimen, then you visualize this bond by some method. Then you can exactly recognize B cells, because only these cells can contain insulin. If you do the same procedure with antibody against glucagon, you will recognize or you will visualize only A cells, and so on. I will speak about immunohistochemical method at the end of our course. There will be one special lecture about it. Electron microscopy can sometimes help to recognize the cells, and it is the case of Langerhans islets. Granules of 4 main types of Langerhans islets differ in their structure. So if you use electron microscopy, you can recognize these 4 main types according to special structure, special character of granules. You can see everything in this picture. Glucagon is situated in granules which are dense core, but their core is eccentric, and remaining matrix of this granule is moderately electron dense. Insulin in B cells is situated in crystalline structures, very dense crystalline structures situated in electron-lucent matrix of a granule. Somatostatin is found in completely electron-dense granules. Pancreatic polypeptide is found in again in dense core granules, but the cores are here now in the center and the surrounding material is electron-lucent. So it is possible to recognize any cell of Langerhans islets in the electron microscope. Only there is a problem with the minor populations like E cells, EC cells, D1 cell, which are very rare but can be present here, and I didn't find specific picture of their granules, so it's probably impossible to recognize them exactly, but they represent really very minor population here. So several examples. Granules of this cell here are very special. It is a granule, it's membrane, and look at the crystalline angular structure inside. It's surely insulin, and so you can say exactly that this cell having granules of this characteristic appearance is a B cell or beta cell. Insulin is containing not only one peptide, but it contains in fact three chains. Two of them, B and A chain, are connected by disulfidic bonds, and they represent the proper insulin. And there is also so-called C peptide, which is longer than both these, or not longer than both, but longer than each of these peptides of the insulin. It represents inactive molecule, but it is important because it can be recognized in blood, and it can be used for the evaluation of the natural ability of pancreas to secrete insulin in patients which are treated by insulin injected. Here, dense core granules, and usually you can see eccentric position of the core, like here for example, or here, and remaining material of the granule is not white, it is very moderately dense. So typical picture of A cell with glucagon, or the last picture of this series, granules which are only black. Their color or their level of electron density is little variable. Nevertheless, whole granules are dense, there aren't any lucent rings, and so we can recognize that here must be somatostatin in these granules, and so you are looking at the D cell here, and so on, so on. Okay, fine. So that is everything about the pancreas and its islets of Langerhans, and I think I can go to liver. Liver is the biggest gland of the body. It is situated in the abdominal cavity, but now intraperitoneally, under the right diaphragmatic copule, and anatomically it is divided into 4 lobes, and histologically it is, or its structure depends on the vascular supply. So let's say something about the vascular supply of the liver. Liver has 2 circulation, in fact, functional and nutritive. Nutritive circulation begins by a hepatic artery, common hepatic artery, which is entering liver and which is bringing oxygenated blood normally from aorta and from the left ventricle. Functional circulation begins by the entrance of portal vein. Portal vein is bringing blood from intestine and spleen, which is rich in nutrients, rich in bilirubin, rich in some waste products. It can be rich in toxins. It depends on the situation. And these two circulation join together at the level of the liver sinusoids. Liver sinusoids are capillaries, atypical capillaries, where both circulations are connected, and at the level of liver sinusoids there is a huge interchange of nutrients, substances, ions and proteins and everything between the blood and tissue of the liver. Then blood goes to venous system, which is beginning by so-called central vein, which then join to form sublobular vein, collecting veins, and at the end hepatic veins, which go to inferior vena cava where circulation of the liver is finished. The same picture contains also the schema of bile ducts, so I'll tell something about it. Bile ducts are beginning in the liver as bile canaliculi. They begin at the same level as are the sinusoids, that means in the hepatic lobule. Then there are several levels of intrahepatic bile ducts which join to form extrahepatic bile ducts, right and left hepatic duct, then common hepatic duct, cystic duct, which is here, and choledochus duct. Cystic ducts goes to gallbladder. Gallbladder is a blind evagination of the duct system or bile duct system, which is important for the storage of bile and for its instantaneous liberation to the duodenum if it is necessary. The liver is encapsulated organ, and as it is intraperitoneally situated, you can find mesothelium on the surface of its capsule. In this case, the mesothelium is composed of simple cuboidal epithelium. It's sometimes possible to find cuboidal epithelium, sometimes cuboidal squamous, simple squamous epithelium can be here. It is variable. Under the mesothelium there is a layer of dense connective tissue, but quite thin, which is then going to the liver as a network of reticular connective tissue. Liver is supported by reticular connective tissue, which serves as its stroma. There are also some islets. If you look at it in section, it looks like islets of loose connective tissue. The amount of loose connective tissue in a human liver isn't high normally, but it can increase in some pathological situations. In healthy human liver, the loose connective tissue is found only about or around vessels and bile ducts, but not directly in the hepatic parenchyma. So the human liver is quite poor in loose connective tissue. The parenchyma of the liver is composed of trabecular epithelium. So after the islets of Langerhans, the liver is the second organ where you can find this special epithelium. Nevertheless, islets, there were microorgans, small collections of epithelial cells, but liver is the biggest gland, so which it is weighing 1.5 kilograms. So it is surely the highest, the biggest the largest collection of the trabecular epithelium in the body. This trabecular epithelium is arranged into the lobules, which are not well recognizable in the human liver due to the absence of connective tissue septa. Nevertheless, I will try to show them in next picture. So, trabecular epithelium, it is the main tissue of the liver, which is supported by reticular connective tissue. Here is a higher magnification of the trabecular epithelium in the liver. It is usually composed of epithelial cell cords, and frequently you can find 2 rows of the epithelial cells in one cord. It is important here because between these cords, like here for example, it's possible to see tiny lines. Here it was visible, or here. These are the smallest bile ducts, which are beginning at the level of the hepatic chords. Or here you can see 2 rows of epithelial cells and very thin line in between, which is representing the beginning of the bile duct system. So trabecular epithelium, usually 2 rows of cells forming cords which are very variably curved, which are anastomosing and dividing, and everything seems to be very irregular. Nevertheless, if you look at it in a small magnification or in this schema, you can see that the overall direction of these cords has some regular course. All these, sorry, I lost my, here it is. All these cords are directing from the periphery of the lobule towards the central part of the lobule. So if you look at the liver in the small magnification, you can see radial organization, radial arrangement of hepatic cords, which are of course not so straight as you can see in this picture, which are anastomosing and disanastomosing, and which are forming a big labyrinth. Nevertheless, they finally tend to go to the center of the lobule. The central structure of each hepatic lobule is a central vein, and so hepatic lobules are sometimes also called lobules of central vein. Here is a schema of one hepatic lobule or one lobule of central vein. Its central vein is this vessel in the center. Now in longitudinal section, hepatic cords are going radially towards this vein, and when we look at the periphery of the lobule, we can find vessels which are supplying our lobule. These vessels situated at the periphery are found in so-called portal space. Portal space is the area where a little bit more connective tissue is present, and where we can find vessels which supply a lobule, and so they are called interlobular. Each portal space can contain interlobular arteries and interlobular veins. And as there is also the bile duct system, which the course of which copies the course of vessels, you can find a third structure in each portal space as well, which is called interlobular bile duct. So portal spaces can usually contain 3 structures: interlobular duct, interlobular artery, and interlobular vein. These interlobular vessels branch to form circumlobular vessels, which go around the lobule. They branch more to the system of sinusoids or precapillary vessels and then sinusoids. At this level, the arterial and venous system anastomose, so the functional and nutritional circulation are connected. As I already said, blood from central vein then goes to sublobular vein, then to collecting vein, hepatic vein, and inferior vena cava. Portal, or also portobiliary space, is the only area in the liver where is some loose connective tissue, and as I already said, you can find usually three structures here: artery, vein, and bile duct. In fact, vessels here are quite small, so maybe better would be to say that you'll find here arteriole, but usually we say artery, which is of course interlobular. Then the bigger structure is interlobular vein, and another small structure but with cuboidal epithelium here is interlobular bile duct. And you can see some connective tissue around, and no other loose connective tissue in the parenchyma of the liver. Of course, there must be reticular connective tissue supporting mainly the sinusoidal endothelial cells. Nevertheless, reticular connective tissue is very bad to visualization in this technique. You must use some special technique like impregnation to see reticular fibers. Here is a higher magnification of some very similar area. These three things found in the portal or portobiliar space can be together called portal triad. So portal triad means interlobular artery, interlobular vein, which is a branch of portal veins, that's why it's P, and interlobular bile duct. Of course, all these structures are branching, so you can find here sometimes some smaller arteries or arterioles, like here, or smaller bile duct, which is just going to our larger interlobular duct. Smaller veins, the proximal segments of circumlobular veins have to be here, and also lymphatic vessels. The author of this picture is labeling this lumen to be a lymphatic vessel. Okay, let's believe, but according to my meaning, it can be also a small vein. Its structure is the same. So portal triad is a typical situation which you can find in the portal space. One vein, one artery, one bile duct, but sometimes more, as you can see here. And now I'll try to show the lobules of liver. This is a picture showing a liver in the not so excellent quality. Nevertheless, I am using this picture to show the lobule here. Yes, here these lines are surrounding or showing borders of one hepatic lobule. This lobule can also be called a lobule of central vein, in Latin lobulus venae centralis. So there must be a central vein in the center. Here I can see a central vein. Now, how to recognize central vein? There isn't any loose connective tissue around it. Also, there isn't any smooth muscle tissue in the wall. It is just a lumen surrounded with endothelium and supported by reticular fibers, nothing more. The same central vein is, for example, here. Another central vein is here. So there are 3 central veins in this picture. So it is like a center of the hepatic lobule. Then we know that hepatic lobule should be hexagonal, usually hexagonal. So we are looking for 6 portal spaces which are present in the corners of this hexagonal shape. Here is one portal space, second portal space. I recognize some connective tissue here as well. So let's say this is the third portal space. Theoretically, another portal space can be here. Here I see something. Here maybe in higher magnification. So you can see that there are not 6 portal space each in one corner of the hexagonal hepatic lobule, but we must try to find the maximum of these spaces. It's a pity that the human lobules are not surrounded by more connective tissue. Theoretically, I can draw another borders of the hepatic lobule, maybe like this. So it can be a lobule with this central vein, or theoretically I can try to delineate this lobule, but I never can be so sure as in the liver of pig, where you can find wait a moment, where you can find perfectly developed septa of loose connective tissue between neighboring hepatic lobule, and so the recognition of lobules in pork liver is much easier than in human. But the amount of connective tissue can increase in human liver in the disease called liver cirrhosis, and when cirrhosis is fully developed, the human liver looks like a pork liver. There are connective tissues around the liver or hepatic lobules. Here is higher magnification of a liver parenchyma, and let's try to recognize borders of one hepatic lobule here. Surely is here central vein. Surely this is portal space with triad, vein, artery, bile duct. Another portal space is here. Another portal space here. Here maybe, so this is the fourth, but in this area I don't find any. Nevertheless, let's omit it. Let's join these portal space which we can see. Let's add some hypothetic portal space which should be here, and we have a hexagonal hepatic lobule. Unfortunately, it isn't possible to do it more exactly. But you can see the tendency of all hepatic chords of the epithelial cells to go towards the central vein, isn't it? This radial arrangement of chords is here quite well visible. Here, this chord, for example, it goes almost straightly towards the vein. Okay, So hepatic lobule, it is the basic morphological unit of the hepatic parenchyme, but not only unit of hepatic parenchyme. Hepatic parenchyme can be also divided into portal lobules. They are important from functional point of view. Portal lobules are triangular structures having 3 central veins in its corners. It is one central vein, second central vein, third central vein, and what is in the center? In the center or near the center of a portal lobule must be a portal space. From the functional point of view, the portal lobule is very important. It is the area of liver parenchyme which is supplied by vessels from one portal space. That means one interlobular vein and one interlobular artery supplies all these structures in the portal lobule, and also bile from all hepatic chords in this triangular area is going towards this portal space and is collected by a bile duct, interlobular bile duct here in the portal space. That's why the portal lobule is also called lobulus venae interlobularis, because it is supplied by one interlobular vein, but also artery and bile duct. So the morphological unit more important from the functional point of view. Let's try to delineate one portal lobule here. I can recognize 3 central veins. There's one central vein, no muscle, no connective tissue around, just endothelium, blood elements inside. Another central vein is here. The third central vein is here. So let's connect them. And here in the center or near the center there is a portal space. I recognize interlobular vein. I recognize here cuboidal cells of interlobular bile duct. Artery is probably this. It is quite small magnification, but I think it can be interlobular artery. So all 3 important structures are here, and portal lobule is clear, I think. Then exists the third unit of the liver parenchyme. It's called portal acinus, or also acinus of venae circumlobularis. It is the area which is between 2 central veins. Here is central vein, here is another central vein. Central vein, and 2 portal spaces or portal spaces are here and here. If you join 2 central veins and 2 neighboring portal spaces, you will get this area. In its center, or virtually in the center here, where is this dashed line, should be one circumlobular vein which is connecting to two portal spaces. I don't know if we can see it here, but maybe circumlobular vein, and these 2 triangles are parts of 2 neighboring hepatic lobules which are supplied by blood from this one circumlobular vein. Probably portal acinus is the most important unit of liver parenchyme from the functional and pathological point of view. I will try to show it in this picture. Here is one central vein. I hope you can recognize the second central vein. One portal space, second portal space, and so this is portal acinus, rectangular structure. And here is the area where circumlobular vein should be, unfortunately not at this level of the section. When you imagine that blood goes from this vein here in the center to both sides through the sinusoids, here near the circumlobular vein should be the first zone where the blood is more oxygenated. Sorry. So here is the oxygenation of blood the best. Here can be a zone where oxygenation of blood is less perfect, so it can be a zone 2, and here near the central vein the oxygenation of blood can be the worst. If you take that these circumlobular vessels contain mixed blood from artery and vein, which is true. The same zones are here in this schema. Zone 1 most oxygenated, zone 2, zone 3 least oxygenated. Normally it doesn't play any role. Oxygenation of blood in liver sinusoids is sufficient for nutrition and oxygenation of all hepatocytes, all parts of the lobule. But there can be pathological situation when the level of oxygen is lower than normally, and in this case this third zone, which is least oxygenated, can be damaged, and it is the case of this picture up. Here is the area of circumlobular vein here. So this is zone 1, zone 2, where hepatic chords seem to be normal. The cells are normally oxygenating, and so they don't suffer from any problems. But here, near the central vein, which is here, you can see vacuolar degeneration of cells, which is a proof of the hypoxic damage. This situation can happen. Opposite problem can be in some intoxications. Toxic damage to classic hepatic lobule can be opposite, because the highest level of this toxin, hypothetic toxin, can be the highest near the circumlobular vein. So near the circumlobular vein, in this zone 1, there are damaged cells, there are damaged cytoplasm, highly eosinophilic necrotic cytoplasm, dispersed nuclei, and on the other hand, here in zone 3, cells are normal, they have light nuclei, normal cytoplasm, because the level of toxins was the highest here in this zone 1, and the cells here absorbed the toxins from blood. So when it came to zone 3, it was the level of toxin was lower and not dangerous for the cells. It is more pathological situation, or only pathological situation. Nevertheless, you can start to study the pathological circumstances of the histological composition of the tissue and organs. Let's go on. I was still speaking about cords of epithelial cells or hepatic cords, and I never said the name of cells which are forming these cords, but I hope you know this name because it is very well known. The cells of the liver cords or hepatic cords are called hepatocytes. Hepatocytes are quite big cells. They can measure about 30 micrometers in diameter, so they are larger than average cells of the body. They have usually one central nucleus, but it isn't exceptional to find cells, hepatocytes with two nuclei, but usually not more. Their cytoplasm is highly eosinophilic because they contain very many mitochondria. Somebody counted each hepatocyte can contain 2000 or 2500 mitochondria, so very many, and that's why the cytoplasm is eosinophilic. Nevertheless, they contain all other organelles. They contain a lot of ribosomes as well, so if they don't contain so many mitochondria, they would be basophilic, but the enormous number of mitochondria is crucial for the total tinction of these cells, of their cytoplasm. They contain also many cellular inclusions. In this picture you can see some lipid droplets in the cytoplasm of hepatocyte. There are here very many. They are very variable in size. They can be present normally here, but if big and too many, they represent pathological sign of steatosis of the liver. The hepatocytes contain also a lot of glycogen. They are able to store sugars, store polysaccharides as glycogen particles, which are usually situated near the nucleus, so the area around the nucleus can be frequently lighter than surrounding cytoplasm because it can contain glycogen. The metabolism of hepatocytes is very active and very variable, so you can find really all structures which were described in cytology, or almost all structures and organelles and inclusions which were described in cytology you can find in the hepatocyte. Hepatocytes are epithelial cells, so they are joined by junctional complexes. They are joined by desmosomes and tight junctions. You can find these desmosomes and tight junction in these borders where hepatocytes are connected together. So these borders or these surfaces can be taken as lateral surfaces. The hepatocytes or the cords of hepatocytes are surrounded by sinusoids. So the surface at the border of hepatocyte and sinusoid, like this one or this one, can be taken as a basal surface of hepatocytes. So we already know basal and lateral surfaces of hepatocytes, and you can maybe ask, where is the apical surface? Can it be here? I must say that yes, it can be here. I will show it in higher magnification. Apical surface is very small, and maybe here I can see it. It is found at these places where two hepatocytes join together by their lateral surfaces. In the middle of the lateral surface is a small tubule, small canaliculus called Bile Canaliculus, where the cells send small microvilli, so it can be taken as a real apical surface of these cells. Let's look at the outer structure of hepatocytes. If you look at the high magnification under the light microscope, you can see that the cytoplasm isn't so much homogeneous as it was in the smaller magnification. We can see eosinophilic grains representing individual mitochondria. Of course, nucleus is every time present if present at the level of the section. Nucleus is light with nucleolus, so typical nucleus of active, very active cell. If you want to find organelles, you can find all of them, as I already said. So a lot of mitochondria, a lot of rough endoplasmic reticulum, cisternae. Okay, they are probably not so many as in serous cell, but the level of protein secretion in hepatocyte is quite high. They have to synthesize and secrete all plasma proteins you can find in blood, all albumin, fibrinogen, prothrombin, some globulins. All these proteins are products of hepatocytes. These proteins are synthesized in rough endoplasmic reticulum, then processed in Golgi complex, here it is, which must be prominent as well, and then secreted using some secretory vesicles on the basal surface. Here is the basal surface, which is situated in the site where the cell is connecting the sinusoidal capillary, sinusoid, and proteins are secreted by vesicles in this direction. They go through endothelial fenestrations or through intercellular spaces in the endothelium, which are quite free into the lumen of sinusoidal capillary, so into blood. So the production of plasma proteins is in fact endocrine production, endocrine secretion of these cells. These cells can produce another products like bile acids by smooth endoplasmic reticulum, SER is here as well, and these products are mostly secreted into the bile canaliculi, which represent this already mentioned small apical surface of these cells. These bile canaliculi are protected by tight junctions, so nothing can leave them. The cell or intercellular space here at this level is very well sealed by tight junctions, and you can see that the lateral surfaces are connected by desmosomes as well. So if I like to repeat the surfaces of the cell, so here is a lateral surface, here is small apical surface with microvilli, and here is quite big basal surface of the cell, which is also equipped by many processes which are called in this picture microvilli, but in fact they are not real microvilli. They are morphologically very similar to microvilli, but they don't go to free space. They go to so-called space of Disse, which isn't free, which contains reticular fibers and reticular cells of the stroma, sometimes, and other cells as well. So for me it isn't easy to say that these processes on the basal surface of a hepatocyte are microvilli. Nevertheless, many textbooks, including Junckera's textbook, use this term, so I will use it as well. But have on mind that microvilli are normally found in apical surface, so these here in the Bile Canaliculus are right or real microvilli, and these processes here are only microvillus-like but not real microvilli. What else can we see in the cytoplasm of this cell? Lipid droplet is here. Lipid droplet, of course, okay. Glycogen situated near the nucleus. Okay, that's everything I said. Lysosomes. Lysosomes are very important. What is the main function of the hepatocyte? Of course, digestion of nutrients. Digestion of nutrients is the main function. So lysosomes must be very, very prominent here. So everything. Peroxisomes are also present here because digestion needs sometimes oxygenation, which is the function of peroxisomes. Okay, a picture from the liver electron micrograph of a hepatocyte. Here is a border between two hepatocytes. Here it's losing, no, maybe not so much. And what can we see here is desmosome. So it is these are two lateral surfaces of two neighboring hepatocytes which are connected by desmosomes. As in other epithelium, I recognize mitochondria, I recognize RAF, endoplasmic reticulum, I can recognize some smooth cisternae of Golgi apparatus, I can recognize glycogen, very highly electron dense clusters, and you can see that there are very many here. I can recognize, no, mitochondria were already said. So yes, okay. The hepatocytes can not only synthesize and store glycogen, but it can also degrade glycogen and quickly liberate glucose back to blood. So the metabolism of glucose and glycogen is very important here, and it is the case of, or it is the function of smooth endoplasmic reticulum, one of the functions of smooth endoplasmic reticulum, which is very important here. It doesn't only produce bile acids, but it also metabolizes glycogen. I was already speaking about protein synthesis and formation of plasma proteins. It was already said. It is the endocrine production of the hepatocyte. Here is the light microscopic proof of glycogen. You can use several methods. One of them is best carmine. Carmine is a red color which is binding the polysaccharides, and so we can see red clusters in the cytoplasm of hepatocytes around one portal space. It is quite nice portal space containing vein, bile duct again, and artery. Electron microscopy focused on glycogen. So the same glycogen which was red in the previous picture is now electron dense and composed of clusters of particles. Mitochondria again, RAF, endoplasmic reticulum. I think that everything here was already demonstrated on the previous picture. Bile acids are synthesized by smooth endoplasmic reticulum, but they can also be recirculated. That means they are reabsorbed in the intestine and go back from blood to the hepatocytes and are directly transported to bile canaliculi. Bile acids are very important components of bile, which are necessary for the emulsification of lipids before they start to be digested by lipases. So that's why smooth endoplasmic reticulum, which wasn't visible in previous small magnification electron micrographs, is here very prominent. This high magnification can show it perfectly. Okay, the same smooth endoplasmic reticulum is also important for bilirubin metabolism. Bilirubin is a product of hemoglobin, degradation, which normally has place mainly in the spleen. So the bilirubin comes by venous blood from spleen to the portal vein and so to the liver. This bilirubin is transported into the cytoplasm of hepatocytes, and this hemoglobin coming from the spleen is water insoluble because it is not conjugated with glucuronic acid. The smooth endoplasmic reticulum of the hepatocyte has a special enzyme which is called Glucoronal Transferase, and this enzyme binds glucuronic acid and bilirubin to make it water soluble, and so it becomes excretable to bile, and so the excess of bilirubin is normally secreted to bile and then transported to intestine and to stool and excreted. Of course, the metabolism of bilirubin has to be very perfect. Mostly, or metabolism of hemoglobin has to be very perfect because the ions, which are parts of the hemoglobin, has to be stored. Iron is very rare element in the human body, and the metabolism of this has to be very storing. We have focused on the hepatocytes so far, but the same pictures contained hepatic sinusoids as well. So let's focus on the hepatic sinusoids now. Hepatic sinusoids are special capillaries or atypical capillaries, which means they are bigger, their lumen is bigger than the lumen of typical capillaries. Normal typical capillaries has got less than 10 micrometers in diameter. Sinusoid can measure 40 micrometers in diameter, and so it can contain many erythrocytes. Blood is flowing through the not only hepatic but through all sinusoids very slowly, and the wall of the sinusoids is composed of very reduced endothelium. The endothelial cells are present here, but they are not well joined by intercellular junctions. They are not supported by continuous basal lamina. Basal lamina is very discontinuous or almost doesn't exist at all, and so the contact of blood and surrounding tissue is very tight here, and the transport of substances between tissue and blood can be very good and fast, because there are many mechanisms of this slow movement of blood, no basal lamina, the poorly developed intercellular junctions, and even more, there are fenestrations and pores in the lateral processes of endothelial cells, so the communication of blood and surrounding tissue and surrounding extracellular space can be very good. All these things are in the liver. Here is a higher magnification. We can see endothelial cell here. Of course, we don't see in light microscope whether there is basal lamina or not, but we will see in the electron microscope that there isn't any basal lamina or only more fragments, and so the communication of blood, which is here, and hepatocyte can be easy through the wall of this endothelium. If you use the electron microscope at first, I'll go back to this schema. You can see what is between the sinusoidal endothelial cell and hepatocyte. Between sinusoidal endothelial cell and hepatocyte is almost nothing. There is a small space. Sometimes there can be some remnant of basal lamina, but unfortunately I don't see any in this picture, but theoretically there can be something, some fragment of basal lamina here or here, which doesn't play a role in the intimate contact of the blood and hepatocyte. Sometimes, but I don't see it here, but it will be in one picture then, there can be some reticular fibers composed of collagen type 3. And what we can see here are processes of hepatocyte, which are again labeled MV like microvilli, but as I already said, I don't like it very much. These are processes of the basal surface of a cell. So why microvilli? But their function and composition is the same as microvilli. They increase the surface, and this increase of surface is important for the communication of hepatocyte and blood, not through the body of the endothelial cell, but for example here through the thin lateral process of endothelial cells. They can be bidirectional movement of nutrients, ions, proteins, hormones, lipids, and so on. Here is a small magnification of the same structure, and I'll go back to previous picture. Here is the schema of this situation. So this is a cord of hepatocytes composed of two hepatocytes with Bile Canaliculus or Bile Capillary in the center. Here is the periphery of the hepatocytes, that means their basal surface with many processes, and this is the space between hepatocyte and sinusoidal endothelial cell. This is the endothelial cell, and this space is called space of Disse. Space of Disse is the tiny space between hepatocyte and sinusoid, where processes of the hepatocyte are present, where reticular fibers are present, where sometimes special Kupfer cells can be present. I will tell what does it mean in a moment. And another type present here, not only Kupfer cells but also Ito cells can be here. Two types of cells can be present in the space of Disse. And this is the space through which all the substances can pass or must pass when transported between hepatocyte and blood. This is a nice picture showing the luminal surface of hepatic sinusoid. Look at these many perforations. These are pores or fenestrations in the lateral processes of the sinusoidal endothelial cells. The only solid areas of the wall are probably areas where are nuclei of the endothelial cells, but lateral processes are perforated like emmental and so the communication of blood and surrounding this space is, as I already said, easy and almost free. High magnification is showing this. You can see pore in this place. Space of this full of processes of hepatocytes. Nevertheless, everything can penetrate through this, and these processes only increase the surface area of the hepatocytes to improve the communication, to improve the ability to absorb everything or secrete everything by the cell. And I promise that I will show a proof that there are reticular fibers in the space of this. When we use impregnation of a liver, we can find reticular fibers situated especially around cords and sinusoids, or between cords and sinusoids. That means in the spaces of this. Now, two cells which can be found in the space of this. The first of them is Kupfer cell. Kupfer cell is here. Kupfer cell is in fact macrophage. You already know several macrophages and several special locations of macrophages. So one of them is a space of this in the liver, and that's why these macrophages have special name. Kupfer cells can be sometimes in the space of this, but sometimes they can be, let's say, intercalated in between the endothelial cells of the sinusoids, and so in fact a part of the Kupfer cell is then exposed to blood and situated in the lumen of the sinusoid. Here you can see erythrocytes, red blood cells, and this is Kupfer cell, which is here in the wall of the sinusoids instead of one endothelial cell. So Kupfer cells can be between hepatocyte and sinusoid. So theoretically Kupfer cell can be here. It was visible in one previous schema, or here as a part of a wall of the sinusoid. They are macrophages, so they phagocytose everything necessary. They can phagocytose any particles coming by blood from the intestine or from the spleen. They can phagocytose old red blood cells together with the spleen or splenic macrophages. You probably know that it is possible to survive a loss of spleen, for example, in some injuries that is necessary to remove spleen, because there can be very serious bleeding, and liver can adapt to this situation, and they can work as a main organ, reducing a number of erythrocytes instead of the spleen. Ito cells are called also hepatic stellate cells, as is here written. They are not so many in the liver, and when filled with lipid droplets, they are recognizable. It is easy to recognize them. They are storing lipids. They are also storing some vitamins which are dissolved in lipids, like vitamin A. But sometimes the amount of lipids is not so high, and in this case these cells become more contractile cells. They are able to produce extracellular matrix, and they can be activated by inflammation, and all these features are important for the development of already mentioned cirrhosis of the liver. Ito cells are probably responsible for the cirrhosis, for the production of extracellular matrix, for the activation of the cells during some inflammation. Cirrhosis is frequently a result of the previous hepatitis, inflammation. Mostly chronic hepatitis can lead into cirrhosis. But also alcohol consumption is activating stellate cells to produce extracellular matrix, and it is also one of the reasons of the cirrhosis. Okay, I can show stellate cell. Of course, in this case it has a big lipid droplet, so it is easy to recognize it. There are also some smaller lipid droplets. Nevertheless, if the lipid droplets aren't here, it would be difficult to recognize it. And there is also Kupfer cell. In this case, it is protruding the lumen of hepatic sinusoid, so it is here included in the endothelium of the sinusoid. Another Kupfer cells are in this picture. In this light microscopic picture, it is difficult to recognize whether, for example, this Kupfer cell here in this part is situated in the space of this or in the sinusoid. It is not easy to recognize. Nevertheless, if you look at this schema here, you can see how the Kupfer cell can be situated really directly in the lumen of the fenestrated endothelium of sinusoid. When we want to really well visualize Kupfer cells, we can use intravital injection of some material which can be phagocytosed. For example, Indian ink is used in this case, and then Kupfer cells and all another macrophages, so it is also found in the spleen or in lungs and in another places where connective tissue is situated. Macrophages can phagocytose this ink, and they become then black, and so when you see black cells in the liver, you can be sure that they are Kupfer cells. But again, without electron microscopy, it isn't easy to recognize whether this cell is in the sinusoid or near the sinusoid. Okay, the last part of this lecture is a system of bile ducts, and also gallbladder will be here. Bile duct system is starting in the liver by bile canaliculi. They are also called bile capillaries or intercellular bile ducts. Maybe this last is the best. Intercellular bile ducts is maybe the best name because it explains where they are present. They are found really between cells, really intercellularly. They are not surrounded by any other things, any other tissues, any other components than by a cytoplasm of hepatocytes. They are situated between hepatocytes, one hepatocyte, second hepatocyte, and between them is the intercellular bile duct. They are sealed by tight junctions, so bile which is secreted into them from the hepatocyte doesn't leak into the intercellular space, doesn't leak through this intercellular space back to space of tissue, for example. And these bile canaliculi are joining together, joining, gradually increasing, and suddenly they got their epithelial lining and they become bile canals of herring. Bile canals of herring are the smallest intrahepatic bile ducts with their own epithelial lining. The cells which are lining not only canals of herrings but all bile ducts are called cholangiocytes. So cholangiocytes are at first flat, then cuboidal, and in the biggest ducts columnar epithelial cells which are lining ducts. Canals of herrings are joining, increasing, and when they are surrounded by connective tissue, they are situated in the portal space, and the bile duct having cuboidal epithelium, simple cuboidal epithelium, is called interlobular ducts. So there were intercellular bile ducts, then canal of herrings, then interlobular ducts, also called bile ductus, but I prefer these terms intercellular and in this case interlobular bile ducts. They are here together with interlobular vein and interlobular artery, which we already know. So let's look at them. This is the picture which I already used when I demonstrated hepatocytes and sinusoids, and now I'll focus to bile capillaries, bile canaliculi, or intercellular bile ducts. They are, for example, here, or here in the cross section, or here when I can show something without label, or here the section becomes oblique, or here there is almost longitudinal section of a bile duct. Yeah, very small, they measure about 1 micrometer. By the way, this is the hepatocyte with 2 nuclei here, as I already said that they can be, but it isn't important at this moment. They are small spaces, they measure about 1 micrometer. When you look at them in the electron microscope, you can see some microvilli inside and tight junctions sealing them. I skipped a picture which is proof or histochemical proof of alkaline phosphatase. Microvilli of the bile ducts are equipped, or their membrane is equipped by alkaline phosphatase, special enzyme which can be well recognized by histochemical methods, and so this method can visualize the system of bile canaliculi maybe better than simple light microscopy without any special staining, but of course much better is the electron microscopy, which is showing everything very well, space, and you can imagine that this smooth endoplasmic reticulum which is here around in the cytoplasm of the hepatocyte is sending here, for example, bile acids or cholesterol or conjugated bilirubin. All these substances then are situated in bile, of course together with water, some ions. Here is a section of canal of herring. You can see the smallest bile duct having its epithelial lining. Epithelial cells are here still very small, squamous or cuboidal, and they are called cholangiocytes. Here the right picture shows electron micrograph. You can see really tube composed of, in this case, almost cuboidal cholangiocytes. These canals of herring join together, they go to portal space, and this is the same picture. They go to portal space and their lining, their cholangiocytes become really cuboidal. Interlobular bile ducts found in the portal space can be recognized according to perfectly cuboidal, simple cuboidal epithelium. They sometimes resemble necklace composed of pearls. Of course, this is cross section. Here is oblique section of such a bile duct, but every time it has a simple cuboidal epithelium. These small interlobular bile ducts then join together to form larger intrahepatic bile ducts having simple columnar epithelium. They join to leave a liver as right and left hepatic ducts, which join to common hepatic duct, and the blind evagination of the cystic duct is a gallbladder. I will demonstrate the structure of all extrahepatic bile ducts in the wall of the gallbladder. Unfortunately, I haven't got any picture of another bile duct, but the wall has the same composition. Gallbladder and extrahepatic bile ducts are hollow organs having a wall composed of 3 layers. There are 3 layers of the wall, so it is the reduced number of layers compared to the main stream of the digestive tract. There are 4 layers. 3 layers of the gallbladder and extrahepatic bile ducts are Tunica Mucosa, Tunica Muscularis, and Tunica serosa. Tunica Mucosa, Tunica Muscularis, and Tunica serosa. So there isn't any Thalassae mucosa. And also, Tunica Mucosa is not so complicated as in intestine or stomach because it has just 2 layers. Tunica Mucosa is composed of epithelial layer and lamina propria, and there isn't any lamina muscularis. The inner surface of Tunica Mucosa is very highly folded, you can see it. There are folds which are sometimes very high. They are anastomosing together to form blind pockets, and these pockets can be sometimes very deep and sometimes can penetrate through the other layers deeper to the wall of the gallbladder. I will show one picture then. The epithelium on the surface of all these folds is simple columnar and very high. It is the highest, but now I can see it. It is the highest simple epithelium in the body. It can measure about 50 micrometers in height, so really very high. It is thicker epithelium than some stratified epithelia. It's very interesting. But before I say something more about this epithelium, let's look at the other layers. Tunica Mucosa, highly folded with high epithelium, lamina propria mucosae. It usually doesn't contain any glands, only near the neck or in the area of the neck of gallbladder there are some mucous glands, simple tubular mucous glands. The Tunica Muscularis is composed of smooth muscle tissue which is mostly circularly oriented. Smooth muscle tissue is here. Then is very thick Tunica serosa. You can see that it contains lot of structures. It contains large vessels. Here is vein, artery. It contains thick collagen fibers. Here it's this area seems to be almost dense connective tissue, but here suddenly is adipose tissue, and it was thicker than the picture, so the next part of this serosa is here. At the outer surface there is mesothelium, because the gallbladder is intraperitoneal organ, so it must be covered by this simple squamous epithelium. As the serosa, tunica serosa, is so thick here, it can be divided into the proper serosa, which is this tissue, mesothelium, and this so-called serous membrane. So this can be taken as a proper serosa, and the inner layers like this adipose tissue and this plate of collagen fibers and many vessels here can be called subserosa layer. So according to some textbooks, the tunica serosa of the gallbladder is divided into the proper serosa and subserosa because it's very, very thick. Once more, these mucosal folds and this high epithelium. This picture from the scanning electron microscope can prove that these structures of the mucosa are really folds and not villi. If you look at them in section, they can resemble villi, you know, from the intestine. But if you look at the scanning electron microscope, the problem is immediately solved. They are not villi, they are flat, continuous folds. Yeah. Okay. I said that the invaginations of the mucosal epithelium can sometimes penetrate very deep. They can penetrate through the muscle layer to serosa, even to serosa. Here you can see a section of a tube which is lined with cholangiocytes, which is lined with the epithelium of the surface. These deep invaginations of mucosa are called Rokitansky-Eschhoff sinuses. According to some authors, they are pathological findings, but they are so frequent in older persons that they can be taken as physiological. So Rokitansky-Eschhoff sinuses frequently found in the slides. Even in our slides you will see them. Several last picture about the epithelium. The epithelium of the gallbladder and other extrahepatic bile ducts is very high. In this high magnification you can see apical protrusions of epithelial cells into the lumen, and you can see that some of these epithelial cells are very light in this apical area. If you look at the scanning electron microscopy, you can see again these bulging apical portions of cells, and if you look at the transmission electron microscopic picture, you can see why the cytoplasm of some these cholangiocytes was so light. There are small granules in the cholangiocytes, and when the granules have got such a light content, they probably contain mucus. So yes, really, cells of the gallbladder epithelium can secrete mucus, not much. It isn't their main function. Their main function is absorption of the bile. Its concentration, that's why they have so many microvilli on the surface, but they also can secrete mucus, which is covering them, protecting them, and so it is important because the substances found in the bile can be sometimes if not toxic, so at least dangerous for for the cells. So the mucus protection is necessary here. I think this is the last picture of this lecture. Yes, really. So thank you for attention. It was my last lecture of this semester. There will be also the second embryological lecture held by Andrea, and that will be everything from us for this time. So wish you good luck in the examination period. I hope you will pass your examinations and also our tests and anatomical tests, and will meet well and in a good condition at the beginning of the second semester. So goodbye.